Hi y'all, this is Mr. Labonte. I'm going to be talking about biomolecules, their structure and function. So what we're going to look at first is carbohydrates. So we're going to look at simple carbohydrates. This one might be saying, I think, therefore I am. And simple carbohydrates. Flowers are pretty. Okay, that's hilarious. All right. But before we even go to carbohydrates, it's essential to understand this concept of a monomer and a polymer. And we're going to be looking at this for carbs, proteins, and nucleic acids. All three of these biomolecules fit into this category. The one that does not fit into this category is fats. So carbs, proteins, and nucleic acids all fit into this concept of monomer polymer. And so what a monomer is it's just one individual example of the thing. But this monomer can be joined with a bunch of other individual examples, and that will become a polymer. So you have a lot of them together. This joining is what's done via what's called dehydration synthesis, and we'll talk about this a little bit more in class. But what I want you to know right now is that if you have one individual, it's a monomer. More than one, generally it's called a polymer. There's a couple of other language things to understand. So you may be saying, well, what exactly are you talking about? In class, we've already looked at one sugar molecule, right? So C6H12O6 is glucose. This would be a monomer. But I might put 4 glucoses together, so 4 C6H12O6s or 16 C6H12O6s, and then that would make it a polymer. So many of them together make it a polymer. All right, so as I've indicated a little bit, there's 4 basic types, and this presentation is going to go over the first type, which is carbohydrates. And the name actually tells you the structure. So carbo, you think carbon. Hydrate, you think water. You see that CHO. Also, actually, the way it should be written is CH2O, right? And so for glucose, the one that we've looked at so far, that's actually 6CH2O, right? And then the 6 ends up being C6H12O6 when you distribute the number, multiplying it by each of those. So this is the one that we're going to look at, but the other ones are fats, proteins, and nucleic acids. And then or you see that you have honk. This is actually just honk, right? HONC in a different order. And then phosphate. But we'll get to that in a later presentation. So starting with carbohydrates, actually we have one more here. Starting with carbohydrates, what you can see is we have a way of describing them. This is a title that my students, or a table that I put in that my students appreciated before. So I'm going to go over this a little bit. So first of all, we have the biomolecule, the basic organization, the specific organization, and an example. We've already talked about glucose. For carbohydrates, you go from monomer to polymer, and then the specific organization is monosaccharide, disaccharide, polysaccharide. So that is if you have one sugar, 2 sugars, more than 2 sugars. And usually at this point I ask a student, how many cupcakes did you eat? And then the student says, I ate 1, 2, many. See, I ate 1 too many cupcakes. 2, not 2. Ha 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 ha. 1 too many. That's the idea. You can have 1 monosaccharide, 2 makes a disaccharide. But then once you get beyond 2, it's just many. So you can just have a number counting system of 1, 2, many. So for carbohydrates, that's what it looks like. And we'll talk about each of these, proteins, nucleic acids, and fats. All right, you have built this in class. So again, NCH2O, this is the basic formula. This is glucose, which we looked at. So this is a straight chain, right? You may remember that that is unstable. And you have 1, 2, 3, 4, 5, 6. So that's what makes it glucose. Pentose, that's 5. You have 5 carbons. Triose is 3. You have 3 carbons. We are not going to look at any 3-carbon molecules, and we're not going to look actually at any pentose molecules as well in this class. We're going to focus just on our 6-carbon molecules. So this is showing you, that highlight on the other one was showing you this as well, but this shows you for glucose, galactose, and fructose, and I definitely am going to want you to know glucose, which we've done, and fructose in terms of their overall structures. What you should see is all of these are C6H12O6. So we're not changing the chemical formula of any of them, but the structure itself does change. And I think it's most easy to see if you're looking at the difference between glucose and fructose. So you can see here, as we did this in class, 1, 2, 3, 4, 5, 6. Those are our 6 carbons, just like the model you made. However, what happens here when you move from fructose to glucose is it ends up having your second carbon be the one that's bonded here, right? So that would have normally been the second carbon. Now we count it as 1, 2, 3, 4. So there's just 4 carbons in this ring, right? And there's an extra carbon on the 1 and an extra carbon on the 4. So that's how you want to think about that. And that actually depends on where the double bond is, if it's on the terminal end or not. Excuse me. So that's what these look like in straight chain form and in their ring form. And I want you simply just to focus on the glucose and fructose. Right? All right. And then we did this in class, remember? Alpha glucose down on first carbon, and beta glucose up on first carbon. That is, when I say down, I mean the OH that's attached to the carbon, and when I say up, I mean the OH that's attached to the carbon. So that's alpha and beta glucose. All right, and now we look at disaccharides. Di means 2. Okay, so this is when you take 2 individual carbon molecules, sugar molecules, and you put them together. So just like before, we'll start over here. This is kind of our most straightforward one. So this is maltose, glucose, and glucose. This is our in this notation you don't actually see the carbons, but there's a carbon there, carbon there, carbon there, carbon, carbon. So that's 1, 2, 3, 4, 5, 6, and 1, 2, 3, 4, 5, 6. Okay, so what I want you to see here when you're looking at glucose in this form that's drawn is you have what's called a 1,4 linkage. And that's because you've got the first carbon here bonding with the fourth carbon via an oxygen. So that's a 1,4 linkage, okay? So this is going to give you maltose. If you've ever had a malt, the sugar that's being used in a malt is maltose. So that's two glucose molecules bonded together. It gets this special name called maltose. The one that you're most familiar with is sucrose. So this is a fructose molecule and a glucose molecule. Please remember though, right, we're still looking at C6H12O6. So even though you have one carbon sitting here and one carbon sitting here, that's exactly the same. And then of course this is still C6H12O6 as well. So, and so this is a 1, 2, 3, 4, again a 1,4 linkage. So you've got a 1,4 linkage there as well. And then finally we have glucose and galactose. These two come together. So this is what a galactose sugar looks like. Just a little bit different in terms of the orientation. This is up, 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 up. So it gets a different name because of the orientation of these OHs. So when, again, a 1,4 linkage, when these two link, we get lactose, and that's the sugar that's in milk. I do not expect you to understand the orientation of galactose. I don't expect you to understand the orientation of fructose unless I specifically tell you that. What I do want you to get with this, though, is that these are all going to be 2 times C6H12O6, right? So these are all just 2 sugar molecules. The chemical equation of them, so like if I did this and I said, this is C12H24O12, the chemical equation is exactly the same for all 3 of these molecules. But as you probably know from the difference between tasting sugar and milk, the taste, the experience of these is significantly different. Milk obviously doesn't just have lactose in it, but the chemical taste is significantly different, and that's because you end up getting these different structures, so that affects your tongue differently. All right, so those are this slide is all about artificial sweeteners and what they look like, and the truth of the matter is we just don't really have enough time to go over all this. With the exception of sucralose, they look very different than what you're used to, but they affect the tongue generally the same amount. So that's it for that one. And now we're going to move on to polysaccharides. So when you put more than 2 glucose or sugars together, any kind of sugars, then you get a polysaccharide. And so what I want you to see, first of all, is that each of those little circles is a sugar molecule. So each one of those little sugar circles is a sugar molecule. And all of these are connected via a 1,4 linkage. But these over here, when it branches like this, this is actually 4,6 linkage. Sorry, I misspoke. It's a 1,6 linkage. And I can draw that in class, but you're not going to need to know that. But you may wonder, why do some of these have branches? All right, so amylose, which is a starch, this only has 1,4 linkages, and that's what gives it its shape. But then glycogen and amylopectin, like if you've ever made jelly or anything, you use this thing called pectin. That's what that is, amylopectin. These have these 1,4 linkages. So these are very, very big molecules, lots and lots of sugars that are in them. So how is it that we get to see this in the body? And so I thought that would be nice for you to see this. So when we eat food, particularly when we eat sugars, our body's going to break up complex carbohydrates and starches down into simple sugars. The simple sugars go into our blood. They then go, sorry about that. These sugars then go into our liver. And our liver converts these simple sugars into glycogen. So the super important task of taking, you can see that is one individual sugar and connecting it to another sugar, another sugar, another sugar, another sugar. This is what happens in our liver. There's a protein in the middle of that glycogen, but for our purposes, you can just think of glycogen as being a bunch of sugars that are added together. And then this glycogen molecule floats around in our blood and then is gradually torn apart, and each of these sugars are used. Sorry about the twisted screen. All right, so how does this work? We'll talk a little bit right here about dehydration synthesis. So if you're looking at, so this is glucose. These are two alpha glucoses that are going to be bonding. And what happens is remember we have alpha glucose, you have down, down, up, down. So that's down. So on the first carbon, you have this OH, and you have this H. These molecules bond together and leave as H2O. And when that happens, you end up with a carbon bonded to an oxygen, bonded to a carbon, and that's the 1,4 linkage right there. So that's what occurs, and this is called dehydration synthesis. All right, the other thing that this slide shows you, and this is, we went over this a little in class when we made it, is beta glucose looks different, right? So because this OH is up here instead of down on the first carbon, and because it's down on the fourth carbon, what has to happen is every other sugar, every other beta glucose sugar has to be upside down. You see that the oxygen is pointing there. And the fact that that's upside down, that's what allows to have these bonds to make wood. Dehydration synthesis still occurs. It's still a 1,4 bond. And this shows you that in just a little more detail. If you look over here, because you have the 6 carbon pointing up in one, and then down in another, it allows these 6 carbons to bond with the 3 carbons like this. That's what makes cellulose, and that's what makes these sugars inedible, because these chains then form bonds between each other. These bonds between each other make these microfibrils that make cellulose fiber. And that's what makes cell walls and the wood in your house. And the only thing that I know of that is capable of digesting cellulose is the bacteria that's inside termite stomachs. So this is a huge amount of food, as we'd call it, so energy, but as we know from the wildfires, but it is not accessible to any but just very unique bacteria. And that's different than glucose, alpha glucose, which is accessible in lots of different ways. Okay, so that is the end of this presentation. I'm going to click to see if I can add a blank page. Okay, so we have, I'm just going to do a quick review. We have biomolecules. And the 4 types: carbs, proteins, nucleic acids, and fats. Fats I'm putting over here on the side because all of these biomolecules have an orientation of monomers can get added together to make polymers. And then in our example, we looked at carbs. So when we looked at carbs, the monomer that we looked at was called a saccharide or a sugar. And the one that I want you to know is C6H12O6. So that's the monomer. And then for the polymer, we have disaccharides. That's a D, sorry. And poly, which is just many. So polysaccharides. So this is the overall organization of monomers and polymers. And then I showed you how they can be combined. So I gave you a list of 3 different disaccharides, right? Maltose, sucrose, and lactose. And so those were the 3 that I gave you. And then I showed you how you can have starches. And the one that I focused on was glycogen. And I focused on cellulose.